hormone. So you should be seeing where previous lectures start plugging into other lectures. You should start seeing the network of information starting to build up at this point. This is where we start seeing systems, start applying all our information. But no pathway of information would be complete without a feedback loop. And so endocrine systems have multiple feedback loops. So here's the same growth pathway. No, actually, this is not. Sorry, this is a different growth. This is this is a different pathway. This is the pathway to release cortisol. Cortisol is generally well, it's used for several things, but it's generally known as a stress hormone. And it starts by the hypothalamus putting out a hormone called corticotropin-releasing hormone, which then targets the anterior pituitary gland, which then puts out the adrenocorticotropic hormone, which will then target the adrenal gland, which will put out cortisol. And cortisol will then target various tissues to create response. But you notice here, there is the feedback. And it goes to multiple places. The cortisol not only feeds back onto the hypothalamus, but it also feeds back onto the anterior pituitary gland. And so we have multiple forms of negative feedback. And so as you can see here, in this name here, we underlined here, we have long loop negative feedback. And you're going to get the name of long loop. It has to go a long distance to make that feedback, negative feedback. And so the idea here is we're taking a hormone and using that as what's going to negatively inhibit the hypothalamus. Now it doesn't just go to the hypothalamus, it also goes to the anterior pituitary gland. And at this point, this loop is called short loop, because it's not going as far back up the pathway to inhibit. Now you can have something called ultra short, where the cortisol inhibits the organ that makes it, but that doesn't happen in every system. More often than not, every system will have a short loop and a long loop negative feedback. Now, how is this different than a normal reflex loop? We've been looking at a lot of reflex loops. For instance, we looked at the parathyroid reflex loop that basically controlled calcium levels. How is this feedback loop different than that one? Look at what's regulating what. In this loop, what's doing the negative feedback? Cortisol, right? In the parathyroid example, what's doing the feedback? The response. What's part of the response specifically?
Is it TSH?
No, there's no TSH.
Did you say it was a thyroid?
Parathyroid.
Parathyroid.
The example is parathyroid and balancing calcium levels. It was calcium that did the negative feedback, right? And calcium was part of the response. Is cortisol part of the response here? No, right? The response is right here. That's not cortisol's not the response. Why is the response here not doing the negative feedback? What's different here? No. The response here is much more abstract. Take the example of growth. Let's take the growth pathway here. There's negative feedback in the growth pathway, right? Growth is a much more abstract variable. In calcium levels is very concrete. There's calcium ions in the blood. There's a concentration to that can be moved up and down. It's a very concrete thing to measure, right? Growth, not a very concrete thing to measure. It's a very abstract, it's a very system-wide response. It's hard to make that into a negative feedback loop, right? It's hard to have that be a signal that will then tell the hypothalamus, stop doing what you're doing, right? So the endocrine system, instead of using the response, uses cortisol, which is essentially the output signal, right? If you remember your feedback loops, right? You have your output signal, you have your target, and then you have your response, right? we're using this output signal now to be our negative feedback here. And that's because that's a much more concrete thing to use as a negative feedback. That has a quantity, it can be measured, it has a concentration. Growth on a whole body scale, much harder to quantify, much harder to use to create negative feedback.
I have a question.
Yeah.
Does cortisol still work in the same way, whereas like if there's too much of it, then the too much, like the excess, will inhibit the anterior pituitary and then
Exactly,
ultimately
right?
hypothalamus?
Just like any negative feedback, as
Right.
cortisol increases in concentration, its negative feedback kind of force, pressure, will also increase and it'll inhibit more effectively the anterior pituitary gland right here and the hypothalamus. In turn, that will, reduce the production of CRH, will then reduce the production of ACTH, will then eventually reduce the production of cortisol. Just like any feedback loop. So yeah. Any other questions? All right. Moving on, we have a lot to cover in a short amount of time. All right, let's talk about hormone interactions. So you can have hormones that do pretty similar things, like glucagon. Glucagon, as you hopefully know from previous lectures, increases blood glucose levels. But cortisol also does it too. The stress response also increases glucose levels, and epinephrine. Does anyone know what epinephrine is? Adrenaline, right? Makes sense that if you're pumping adrenaline to the body, you also need to have some extra glucose circulating around so you can do your fight or flight response, right? So epinephrine also can increase the blood glucose levels. Well, if you have 3 different hormones doing it, what happens when multiple of them are in the bloodstream at the same time? You can have synergistic effects, exactly. And so this is what the graph on the right-hand side is showing. These are synergistic effects. So here are these lines showing the effect of blood glucose levels in the blood over time. And so you have these various responses over time. And the thing about this is this is called synergistic or synergism. When you take 2 hormones and you put them both in the bloodstream and you have them both create a response, they create a response that's greater than the sum of the 2 parts individually. So 1 plus 2 is now greater than 3, is the pseudo-math behind this. In the sense that if you combined epinephrine and, let's say, glucagon, if you add those 2 levels together, it is still lower than what is the observed real effect. There is some synergy that when you add them together, you get more than the effect that if you just added the 2 things independently. Does that make sense? This kind of has this somewhat multiplicative effect in a way. And notice if you add all these 3 hormones together, you can see like there's a tick here, there's a tick here, so that's 1 plus 1, 2 plus 1, 2, 3. That's 6 ticks. 1, 2, 3, 4, 5, 6. Mathematically, if you add all 3 together, you get somewhere around here. But we see you can get much, much, much higher if you had all 3 in play. Yes?
Is the graph accurate?
Yeah.
5 hours, it would spike after 5 hours.
Mm-hmm.
So would 5 hours
Cortisol
be the
and
halfway?
glucagon are long-term. Cortisol is a steroid hormone, so that actually lasts a long time. And glucagon is usually produced on the hour scale because it usually involves, energy balance. Epinephrine, granted, is not used in hours, but if induced for hours, can have this effect. Now, it's not 100% clear why this comes upon. What is the real mechanism of synergism in hormones? Some researchers believe it's effective, kind of an amplification effect, that you're making a lot more second messengers in these target cells than you would have with each of these hormones separately. And so there's perhaps some amplification of the amplification, so to speak, in the signal transduction. That is the current idea right now, but it's not a very solid idea. It's just the best guess we have right now. You can have other hormone interactions. you can have something called permissive interactions. This is kind of the idea of one hormone being the gatekeeper hormone. And so, for example, let's look at sexual development. This is the same example in your book. If you have hormone A, it doesn't work by itself. This is hormone A, and this is time, and the x-axis, it would just look like this. It would not by itself would not induce any sexual development, right? But hormone B works on itself, but not very well. Meaning, it can eventually give rise to sexual development, but at a much later point. It's only if you had A and B together. So in this example, it's the thyroid hormone and the sex hormone together. Where do you see that is supposed to be a different color? Sorry. Where you see normal sexual development. So this is the idea that this is permissive. It's not that they're synergistic, not like they're making an amplified effect. It's just the fact that one hormone is required for the other hormone to work properly. The last type of interaction is antagonistic, or opposing hormones. So there are two hormones that do opposite effects, and hopefully you remember the previous example of insulin and glucagon, right? Glucagon raises the blood glucose levels, insulin reduces the blood glucose levels, and they work in tandem. When one is high, the other is low, and vice versa. And so this is an example of antagonistic hormones. And you've seen this example before, so I'm not going to have to really explain it too much. But yeah, those are the three different types of hormone interactions. Let's move on to some